So, today we are talking about protein structure and analysis, specifically within the realms of biotechnology. So, first off, what is protein synthesis? Protein synthesis, right, is our overview. So, what are proteins? Proteins are essential biological molecules made of amino acids, and they play a critical role in various life processes. And those things are things like enzymes, producing hormones, carrying things to and from cells, and so on. So, each protein is encoded by a gene, and a gene serves as a template for synthesis of a messenger RNA or mRNA. And mRNA then serves as a template for the synthesis of a polypeptide chain. And then the polypeptide chain is folded to make a functional protein. So this goes back to the central dogma that we've learned about in biology, where DNA makes RNA makes proteins. So we can see that DNA, right, genes, make RNA, make mRNA, and then mRNA makes polypeptide chains, and those are folded into functional proteins. So, we're going to look at bacterial or prokaryotic transcription and eukaryotic transcription. So, bacterial transcription is regulated by proteins that bind to a region upstream of a gene called the promoter region. Translation is initiated by an RNA polymerase binding to the promoter region. So it sticks onto that region. The promoter region says, hey, bind here. It sticks, and then it starts reading the DNA and transcribing it into RNA. So RNA polymerase is an enzyme that generates mRNA molecules. So it acts very similar to the DNA polymerase, except it produces RNA instead of DNA, so it changes that adenine and thymine bond to an adenine and uracil bond. So, steps in bacterial translation. First, RNA polymerase unwinds the DNA. Then RNA polymerase moves 3 to 5 prime, adding ribonucleosides. And the new strand is complementary to the template DNA strand, except A equals U instead of A equals T, like I just talked about. This continues until RNA polymerase reaches a termination sequence, and then the RNA polymerase releases, and the mRNA releases and moves out and away from the DNA to the next step. So eukaryotic transcription is a little bit more complex than bacterial genes. Eukaryotic genes are more complex. They have multiple units called exons and introns. So how I like to remember the difference between exons and introns stay inside the nucleus and exons exit the nucleus. So pre-mRNA, the exon and intron sequences are present, and that pre-mRNA is processed to remove the introns and join the exons together in a process called RNA splicing. Once those exons are spliced together, mRNA is capped with a modified guanine base on the 5' end and a long sequence of adenines called a poly A tail on the 3' end. These additions are thought to protect the mRNA from degradation or falling apart. And once this is all processed, it is considered or called mature mRNA. So here we can see some differences between the two. We can see, right, the location, prokaryotes, transcription and translation occur in the cytoplasm, while eukaryotes we can see in the nucleus, and then move into the cytoplasm for that translation. There is no mRNA processing in prokaryotes, but it is complex in eukaryotes. Regulations is less complex in prokaryotes than it is in eukaryotes, and the ribosomes are smaller in prokaryotes as they are in eukaryotes. So, translation happens when we take the mRNA sequence and translate it or change its language to a sequence of amino acids. We map So we take the mRNA and map units into 3 nucleotides, and these correspond to amino acids. Each nucleotide unit codes for something called a codon. And there are 64 possible codons, and there are only 20 possible amino acids. So multiple codons encode for the same amino acid. So we get a redundancy, which helps reduce errors. mRNA start codon, in most cases, is AUG, which codes for methionine. And then translation ends when it reaches a stop codon. And the stop codon has no corresponding amino acid. The genetic code is shared among all organisms. So, the amino acids are the same in bacteria, plants, and humans. So, to do translation, this is just a quick reminder from biology. Specialized RNA molecules called transfer RNA or tRNA read and translate the genetic code. So it takes mRNA, changes the language into those amino acids. They transcribe, right, the nucleotide sequence on the mRNA to the amino acid sequence. You can kind of see, right, the anticodon here attaches to the mRNA and the acceptor arm, right, where the amino acid connects to. tRNA are just RNA molecules, as you can see here, that have been folded into a 3D structure. So, to go through translation, we do have some ribosomes, and translation occurs at these ribosomes. Ribosomes are 2 subunits, right? They have large and small, and they differ between prokaryotics and eukaryotics, right? I told you that prokaryotics are smaller and less complex, while eukaryotic ones are bigger and more complex. Prokaryotic subunits, the small one is a 30S and the large one is a 40S, while eukaryotic subunits is 40S for the small one and a 60S for a large one. So the large subunit has 3 sites, an A, P, and E site. The A site is the accepting site. So it accepts the amino acid and holds the next tRNA carrying the amino acid sequence. The P site is the polypeptide site, and this holds the tRNA carrying the growing polypeptide chain. And then E is the exit site or the ejection site. So it comes in to the A, moves to the P, and exits through the E. So in translation, there are 3 steps to consider. We have initiation, elongation, and termination. Initiation, translation begins when a small ribosomal subunit binds to the mRNA and charged tRNA binds to the start codon, so the AUG. And then the tRNA carrying the methionine binds to that start codon. Next, the large subunit binds, and then we start adding. So elongation starts when the next RNA comes to the A site. mRNA is moved through the ribosome as the codons are read. Each mRNA codon codes for a specific amino acid, and we use that codon chart that we saw to determine those amino acids. Elongation occurs in steps. Codons recognize the appropriate anticodon in the next tRNA, and that goes to the A site. Polypeptide bonds form and transfer polypeptides from the A site to the P site, right? Translocation, A site moves to the P site, and then the P site goes to the E site, and the A site opens up again. Termination occurs when a stop codon is reached in the A site of the ribosome. The stop codon does not code for an amino acid, and it will signal for a release factor. The bonds holding the polypeptide to the P site are released, and the polypeptide moves along to the next step. All translational units are disassembled and go on to the next one. So the availability of protons to regulate is regulated by the rates of transcription and translation. The activity of proteins is also regulated by modifications made after translation called post-translational modification. So these include the addition of carbohydrate molecules and phosphate groups. So we can see glycerin facilitates the interaction between cells and their environment. So it's very important that we add these post-translational translational modifications to these proteins before they become completely functional. I will see you in the next little section.